Very good evening to one and all. Welcome back to the today's special session. Today we have our guest speaker, Dr. Suraj Singh Bhadoria sir. We welcome you, sir. We welcome you all the participants. And today we'll have the topic regarding nanoparticles in homeopathy medicines. And today we also have our National Vice President, ma'am, Dr. Niveta Patnaik, ma'am. So we welcome you, ma'am. A special welcome to you, ma'am. So I hope this will be a good session regarding the new topic, the nanoparticles in homeopathy medicines. So we welcome you, sir. We welcome you, Dr. Niveta Patnaik, ma'am, also. So we welcome all the participants. So coming on to the introduction regarding the sir, sir has been completed UG in SKP Gujarati Homeopathy Medical College, Indore, and sir did masters in Bharati Vidyapeeth Homeopathy Medical College and Bharati University, Pune. And now sir is pursuing part-time PhD scholar from Vinayaka Mission University, Salem, Tamil Nadu. So glad to be on the board, sir. So the session is all yours, sir.
Okay, first of all, good evening to everyone, to respected madam, and to all my colleagues and all the faculties and the students of this homeopathy. Today, we are going to discuss about nanoparticles. What are nanoparticles? How we will prepare the nanoparticles? What are the different features and the characteristics, the properties of the nanoparticles? And how we will prepare the nanoparticles in homeopathy, and what is the utilization of this minute and very smallest structures. So today, this nanoparticles, it is visible to all, ma'am. Is it visible to all, ma'am?
Yes, sir. Yes, sir.
Okay, ma'am. So basically, a nanoparticle or the ultrafine particle, it is a particle of the matter which lies in between 1 to 100 nanometer in a diameter. So the range of that ultra fine particle is in between 1 to 100 nanometer. And sometimes it is also used to have the large particles which is up to 500 nm. So when we talk about nanoparticles, so in nanometer it lies in between 1 to 100, whereas in micron, when we talk about in micron, in micro, so it lies in between 0 to 1 micron. So it is the very ultra fine, very smallest and very what we can say minute structures which has a fibric or the cubic shape, and it is having a different dimension. It is having a different structures and the pattern of arrangement. So this is a size of nanoparticle. When we talk about the nanoparticle range in different aspect, so nanoparticles, these are microparticles, and they are very fine and very smallest. And the coarse particle, when we compare this minute and the nanoparticle with the coarse particle, so in coarse particle, the range is lies in between 2500 to 10,000 nanometer. So this is the size of coarse particle. Because in our pharmaceutical practices, when we talk about drug, when we talk about medicine, when we talk about any product, any medicinal product and any remedy, so the coarse particle, they are bigger in size. They are not micro and minute in structure. So the range of this coarse particle is lies in between 2500 to 10,000 nanometer, whereas this nanoparticle, the size is very minute. They are very smallest and they are just like a very fine structures. So these are the differences between the size of nanoparticle as compared to the coarse particle. Suppose when we talk about cluster, suppose when we talk about colloidal particles, so they are coarse particles. That's why they are not easily filtered out, and that's why they are not easily miscible in any solution. But these nanoparticles, their miscibility is very higher and very what we can say fast as compared to the other coarse particle. their rate of absorption is also faster than the coarse particle. That's why today we shifted towards nanoparticle. Now we have nanomedicine. Why? So the rate of absorption and the rate of miscibility of this particle, the minute structures, are higher than the coarse particle. Because of their smaller size, they have different physical as well as the chemical property. Clear? So every nanoparticle, every substances, they bear a different physical property and a different chemical property, like colloidal properties, like ultra fast optical effects, like electrical property, a field of electricity. So these are the different aspects and the characteristics of nanoparticles. So it depends on so many things. It depends on the source where we prepare the nanoparticle. It depends on the family. It depends on the class. It depends on the structure of the nanoparticle. It depends on the miscibility. It depends on the mixture of that particular nanoparticle. Either they are simplex variety or the complex variety. So there are various aspects of talking about the physical and the chemical properties of nanoparticle. Moving towards next slide. Now this nanoparticle, they are different in a nature. They are generally have a different dimension in the science of chemistry, in the science of physics, in the science of geology, in the science of biology. And when we talk about the transition phase of this bulky material and the atomic and the molecular structure, this microstructure and the minute particles, these nanoparticles, they are very dynamic in nature. They are not easily visible. They are not having a mass. They are very minute and minute. So that is very different and very important aspect of nanoparticle. And they are a very important component of atmospheric pollution. When we talk about pollution, so in the atmosphere, there are so many carbon nanoparticles. There are so many metallic nanoparticles. So these metallic nanoparticles and the carbon nanoparticles, they are responsible for the certain disease condition or the pathology or which affect the human life or the living entity. Either they are plants, animals, or the lower vertebrates. So this is very important part. And when we talk about the industrialized products such as paints, plastics, metals, ceramics, magnetic product, they all possess nanoparticles. This product and the what we can say the formation of nanoparticle with the specific property like physical property, chemical property, their biological property, their pharmacological property, it comes under the branch of nanotechnology. So this is nanoparticles. So nanotechnology is what? Nanotechnology is the branch of science which deals with the formation, the categorization, study, and the basic properties of the minute structure, which are nanoparticles. What is the range? In nanometer, it lies in between what we can say. हमने पहले पढ़ा है प्रीवियस स्लाइड में. It lies in between 1 to 100. ठीक है. And when we talk about in micron, so it lies in between 0 to 1 micron. So this is the range of nanoparticle. According to IUPAC, now it is very important and standard one. When we talk about any material, any particle, and any biomolecular sciences, so the IUPAC, which stands for International Union of Pure and Applied Chemistry, plays a major role. So in 2012, this IUPAC, they derived this nanoparticles, the polymeric and the very minute structure with the any different shape and the arrangement in 1 into 10 to the power minus 9 and 1 into 10 to the power minus 7 m range. So this is the IUPAC classification. They define such minute and the microstructures in terms of their range, in terms of their division, in terms of their categorization in size or the mass of particle. This definition evolved from one of this association in 1997. So this IUPAC plays a major role. Suppose when we talk about the classification of enzyme, so again it is derived by IUPAC. There are different classes of enzyme like oxidoreductase, transferase, hydrolase, lysase, epimerase, ligase. So IUPAC plays a major role. It is a standard one. It gives you a specific dimension of that molecular and the atomic structure. So it plays a major role. When we talk about the history part, so this nanoparticles, they are generally produced by us different what we can say, the aspect of cosmological, the natural occurrence, geological occurrence, meteorological occurrence, and the biological processes. So these are the different ways through which this minute and the micro, very finest structure derived. And the main significant fraction of this interplanetary dust and this, what we can say, the nanoparticle, they are generally falling on the earth at the rate of thousands of tons per year. So it is the nanoparticle range. So similarly, when we talk about the atmospheric dust particle, so many viruses have diameters in the nanoparticle range. Suppose when we talk about recently when we had this COVID-19 pandemic, so this coronavirus, their range, it is very minute and very smallest. It lies in between the range of this nanoparticle between 0 to 1 micron, and in nanometer it is around 1 to 10 or 100. So it is very minute. So when the size is less, so the rate of penetration is more. That's why in COVID-19 the fatal rate is more as compared to other viral infection. So when the coarse particle, when the size of the bacteria or the virus is bigger, so the rate of penetration and the rate of fatal condition is less. So it is inversely proportional. Similarly, when we prepare any drug material, so when the size of particle is more, so the rate of absorption is less, so the rate of distribution is less, so the rate of metabolism is less, because it need more and more catalytic action. That's why today in the modern scenario, the allopathy medicine, they are more shifted towards the nano medicine and the specificity. Now they are moving towards homeopathic principles. Now we can't say that they are not following the individualization, they are not following the law of specificity. They are now following the law of specificity, individualization, and the nanoparticles. But ultimately in our homeopathic medicine, our medicines are already ultra high diluted. They are like very less and minute structure. So this is very important part why this nanoparticles plays a major role in ultra high dilution. Why we need to create such mass particles in our ultra high dilution? Because nowadays we have no such evidence based practice. We just talk about water memory, silica hypothesis, plausibility phenomena, epitaxy. But where is the evidence based medicine? So to prove that, now we are going to create some mass in our ultra high dilution. That is our main aim and objective. Moving towards next slide. Now, morphology and the structure of nanoparticles. Nanoparticles are generally having a different variety in the shape, and they are in a different what we can say structure or the pattern of arrangement. Like some are in a rod shape, so they are said to be a nano rod. Some are in a chain, so they are nano chain. Some are in a decahedral nanoparticles, so they have a very different arrangement. Nano stars, they are in a star shape. Nano flowers, so they are in a flower shape. Nano reefs, so they are in a reef shape. Nano ring whiskers, they are in a different arrangement of the particle size. Nano fibers, nano boxes, they have a cubic shape. So these are the different forms and these are the different structures of this minute and the massless particles. When we talk about the shape of nanoparticles, so generally determined by the intrinsic crystalline habits of the material. So generally when we talk about these nanoparticles, so they are generally influenced by the environment, by their creation, because our environment is a heterogeneous in their nature, in their structure. There is no homogeneity. We have a multiple compounds, we have a multiple dimensions, we have a multiple flora. So these all heterogeneity and the multiple components create a different shape and the size and the dimension and the structure of this massless minute nanoparticles. Generally this nanoparticles, they are very sensitive, they are very finest particle. And whenever we talk about this crystalline growth, emulsion and the micelles, so generally they are just a precursor of such preparation. They are not nanoparticle, they are the precursor of that particular massless particle, because ultimately when we talk about digestion, so when we digest this emulsion and micelles and the other crystalline material, so ultimately the size is going to reduce. So they are just having a different aspect of the preparation and the arrangement of particles. So generally these nanoparticles, they are surrounded by certain crystalline growth and the emulsions and some other droplets. So they are just a part of this minute structure. But in some other application of nanoparticles, they have a very specific shape, they have a very specific size, and they have a very specific range. So there is a specificity in the shape, in the size, in the nature, in the pattern, in the arrangement. So because of that specificity, they have a very strong aspect of what we can say the pharmacological action. Because ultimately why we are going to prepare this, because this minute and the finest particles, they are showing more efficacy as compared to the coarse particle. So that's why we are moving towards such minute and the microstructure in terms of therapeutic value. In production, we have a various method through which we prepare such nanostructures, like mechanical breakdown biopolymer method, where which we are going to have the lysis of biopolymers. Pyrolysis, lysis means breakdown. Condensation from the plasma, inert gas condensation, radiolysis, wet chemistry, and the ion implantation. So these are the different methods through which we prepare such minute structures. So by different method, they have a different arrangement of the pattern, their size, their shape, their range, their action, their rate of absorption, rate of penetration. Everything is very different. So it depends on the nature of production. So it is very important part. When we talk about trituration, now where is homeopathy? Suppose when we talk about trituration, so this trituration, it comes under the mechanical process. Okay, so when we do trituration, so ultimately this is a force in acceleration. This force in acceleration accelerate the mass particle into massless particle by changing the valency, number one. Number two, by changing the position, like lower shell to higher shell to lower shell, by making the bond or by breaking the bond. So these are the main aspect of trituration. That's why in our homeopathy we are having, or we have ultra high dilution, we have minute structure, we have micro theory. So this is only because of the mechanical process. So there are some other process through which we are going to produce, we are going to synthesize these nanoparticles in the field of medical science. Characterization, these nanoparticles are different in their analytical requirements than conventional chemicals. for which the chemical composition and the other concentration are sufficient metrics. So they have a different composition, they have a different property, they have a different physical characteristic, they have a different chemical characteristic. Nanoparticles, they are very different and specific in their shape, size, surface properties, because the surface properties plays a major role. Because ultimately when we deal with the surface property, so ultimately this will enhance the action of that massless particle. So this surface property plays a major role. Crystalline nature and the dispersion rate. So these all properties are very important in aspect of nanoparticles. Suppose if we create any massless particle, and if suppose we are not focusing on their surface property, if it is very bigger in size and if it is surrounded and overlap by the multiple particles, so their surface property affects. So they are not able to show their specific action. They are not able to show their deeper penetration or the high rate of absorption or high rate of distribution or high rate of metabolism or the excretion. So surface property plays a major role. Next is now these are the different methods or the tools or the equipments through which we standardize, quantify or qualify the nanoparticles. Now what are the different methods like spectroscopy, electromagnetic radiation, X-ray, ultraviolet rays, UV visible spectrophotometer is one of the devices which can be used to measure the size of this particle and the concentration of this massless particle. Another one is electrophoresis for the surface properties, XRD, X-ray diffractometer or the X-ray diffraction technique, mass spectroscopy, chromatography, centrifugation and filtration. Generally this chromatography, centrifugation and filtration for the coarse particle. But sometimes in preparation of nanoparticles, there are certain turbidity, there are certain immiscibility, there are certain colloidal particles. So to prevent such coarse particle, we are going to filtrate it, we are going to centrifuge it, we are going to have that particular concentration of the compound. So this is only because of the centrifugation, filtration, single, double filtration or the chromatography. So the main aspect is the spectroscopy. Raman spectroscopy also plays a major role. It is discovered by Dr. C.V. Raman. So again, this Raman spectroscopy plays a major role for the standardization of ultra high dilution. Generally, this Raman spectroscopy is based on the phenomena of H and OH bond vibrational frequency. Based on the phenomena of H and OH vibrational bond frequency. H and OH, that is water molecule. So this is Raman spectroscopy. It is having only monochromatic light. Whereas in UV visible spectroscopy, it is having ultraviolet light. In XRD, it is having X-rays. So different spectrometer, different medium or the range of light, they are going to quantify and qualify this particle size and their nature also. So these are the different methods through which we can standardize our material. Another one is NMR, nuclear magnetic resonance. Generally, it detects the magnetic property around the nucleus. So that is the main principle of NMR, that is nuclear magnetic resonance. It detects, identify, it based on the electromagnetic field around the nucleus of atomic structure. So this is NMR. So there are different aspects of standardization of nanoparticles. The another one is, which is not written in this slide, but again it is very important, that is electron microscope. It is having two different range, SEM and TEM, scanning electron microscope and transmission electron microscope. So this electron microscope, it is also indicating the surface property, surface morphology, and the structure, the pattern of arrangement of nanoparticles. These are the various devices which are useful for us, and we have to focus on that part, because ultimately, suppose if we prepare nanoparticle, suppose if it is liquid phase, so either we have SEM or TEM, either we should go for SEM and TEM. So it depends on the nature of solubility, it depends on the nature of solvent, it depends on the nature of the nanoparticle. If it is coarse powder, so SEM is more feasible, scanning electron microscope. If it is liquid sample, then TEM is more feasible, transmission electron microscope. But fortunately, by our homeopathic samples in our Parul University, by our first year students and the PG students, second year BHMS students, they proven the surface morphology of liquid nanoparticle by STEM also. So this is a huge victory of homeopathy, because ultimately we all are the pillar of this homeopathy. So this is a very different aspect of this type of practices. So we have to know about this. Next is, what are the application? Why we prepare this nanoparticle? So it is used for the biomedical aspect. It is important for us for various disease or the target cell and the tissue. Sunscreens, road paving, photocatalytic action, liquid properties turner, and the polymer reinforcement. So these are the different application and the use of nanoparticles. For our pathy, this biomedical plays a major role. Now, moving to us next slide. Now, this is a real image Echinacea nanoparticle in 2 micron and 15 micron. So they have a different pattern of arrangement, and this is Echinacea mother tincture. So it is important. It covered by our first year BHMS students. They completed this research work, not only showcasing this structure, they also proven this structure in vitro and animal model. So this is Echinacea nanoparticle in 2 micron and 15 micron. In 15 micron, you can see just it is just seen like a coarse particle, a bigger, very large shape, which is driven into the sea of water. So these are the different arrangement and the structure and the shape of nanoparticle, which is in Echinacea mother tincture. So it is also standardized by SEM only, scanning electron microscope. In Parul University, we have a micro nano lab in our Parul University. So this micro nano lab, we have SEM, we have AFM, atomic force microscope, we have UV visible spectroscope. Now we are moving towards STEM also. So this is one of the research work completed by first year BHMS student. Now this is Echinacea nanoparticle in mother tincture in 30 μm. Now in 30 μm, you can see this is a small rod-like structure in a cluster shape. So small rod shape of filaments which conjoin together and form a cluster shape, which is having irregular margin. So this is in 30 micron. In 5 micron, you can see a small colloidal particle which is spread over the clustering of the nano size. So this is the Echinacea nanoparticle. So they have a different arrangement in a different range. Now this is Echinacea nanoparticle in 5 micron. So this is just like a bottle, is just a broken bottle or a broken glass like arrangement in a sea. So this is a irregularity and a crystalline nature of the nanoparticle. In 5 micron, you can see a golden appearance, and the golden is appearance of this nanoparticle. So this is Echinacea mother tincture. Moving towards EDS scanning, this EDS give you the compositional value of this nanoparticle. What are the composition of this nanoparticle which seen in Echinacea mother tincture? Now you can see the composition. Carbon in weight percentage, it is around 81.4. Oxygen in weight percentage, it is around 15.7. Potassium in weight percentage, it is around 1.7. Iron Fe in weight percentage, it is around 1.2. The sigma percentage of carbon is 0.7. The sigma percentage of oxygen is 0.6. The sigma percentage of potassium is 0.1. And the sigma percentage of iron Fe is 0.2. So these are the different compositional value of echinacea nanoparticle in a mother tincture, in a hydroalcoholic base, because our mother tincture is a hydroalcoholic base as per the new method. Now again you can see this compositional value in EDS scanning, 81.42 in a carbon, oxygen is around 15.67, potassium is around 1.67, iron is around 1.23. Whenever we talk about the atomic percentage, so the carbon is more as compared to other elements like oxygen, potassium and iron. So carbon is around 86.65 percentage, oxygen is around 12.52 percentage, potassium is around 0.55 percentage, and iron is around 0.28 percentage. What it determines? It determines that this particular nanoparticles, they are having more efficacy in the carbo-nitrogenate constitution. Ultimately, we have a different types of constitution: hydrogenoid, oxygenoid, carbo-nitrogenoid. So in carbo-nitrogenoid, they have more carbon and the nitrogen element. So in our preparation, in our nano formulation, in our nano composition, when the carbon element is more, so definitely that particle is more deviated and moving and showing efficacy towards the carbo-nitrogenoid. When it is having more oxygen, so they are showing more efficacy towards oxygenoid. When it is having more hydrogen and OH bond, so it is having more efficacy towards hydrogenoid constitution. So that is the main aspect of this compositional value of nanoparticle. Why we want this composition? Why we want to know about this? So this is the main aspect of such preparations. Moving towards next slide. Now this is the Azadirachta indica nanoparticle. Now it is Azadirachta indica nanoparticle. It is prepared in the Azadirachta indica mother tincture. Now you can see this is just like a small cubic and the crystalline glass shape, which are in a field of scattering, in a field of light, and they have a different dimension. They have a different patterns of arrangement. They have a different size. So this is Azadirachta indica nanoparticle. This is in 1 micron. Now in 1 micron, you can see this shape of Azadirachta indica nanoparticle in hydroalcoholic base. Now another one, this is sunflower nanoparticle. So this is helianthus. So it is sunflower nanoparticle. So you can see again they have a different shape of arrangement. They have a different pattern of arrangement. They have a different dimension. So these are very minute structures in helianthus cube. This is then again hydroalcoholic base. So different mother tincture, because of different preparation, because of the different source, because of the different family, they shown different pattern of arrangement. They shown different pattern of size. They shown different properties like physical, chemical, and their therapeutic also. So this is a very important characteristic of the nanoparticle. Now this is sunflower nanoparticle, helianthus Q nanoparticle in 2 micron. So you can see this arrangement. In some pattern, you can see the regularity in their material, but in some property you can see the irregularity. See, this is the regularity in the pattern, whereas in this form, this is just like a irregularity in the pattern. So this is the different pattern of arrangement of helianthus Q nanoparticle. Now this is again helianthus Q nanoparticle in 150 micron. So in 150 micron, it is in a bigger shape, it is in a bigger size, just like a piece of stone. But beside of these pieces, now you can see these are the very minute and the microfilaments. Now these are our nanoparticles because it is 150 micron. So it is not nearby the range of nanoparticle. It is above the range of nanoparticle. Now this is our workshop conducted by Micro Nano Department of the Parul University for facilitating the nano technology in various disciplines like agricultural sciences, like homeopathy, like Ayurvedic sciences, like information technology, engineering, chemical branch. So we every time our college and our faculty is going to organize a workshop or the seminar and invite some specialized person to conclude their research, their work, and their knowledge for nourishing the other faculties. So this is our research training specific on STEM, scanning electron microscope in the Parul University. Now this is our student research work. Now this is our main part. As a teacher, what we want to do, we want to nourish our students. We want to nourish our PG students, our PhD students, our bachelor students in a right propagation, in a right direction, with the hard work, with the honesty, with the full of dedication, with the confidence, with the positive attitude. So this is our first year student, Drashti Baria. She prepared a conjoined nanoparticle with the complex variety. What she prepared? She prepared the conjoined nanoparticle by mixing Cantharis and Staphylos aureus mother tincture. So that is their main aim, to prepare the nanoparticle by the complex variety by mixing Cantharis mother tincture and the Staphylos aureus mother tincture in the hydroalcoholic base. And afterwards she had this trituration or the succussion by electric potentizer and the triturator machine. So that is their project based on the complex variety and the conjoint formation of biomolecule. Another one, these are our students of Jawaharlal Nehru Homeopathic Medical College, and they prepared helianthus nanoparticle in hydroalcoholic base, the mortar and pestle. When we do trituration, similarly they started preparing emulsion. So that is their project. So these are our first year students. Now these are our PG students. Now they prepare nanoparticles in a oil base. Generally in oil base, it is very difficult to showcase the minuteness and the microfilaments, the pattern of nanostructures. The density is more, the viscosity is more. But again, they are successfully prepared the nanoparticles. They standardize the nanoparticle by scanning electron microscope, by the electron microscope in our micro nano lab of Parul University. And they also standardize the nanoparticle by UV visible spectrophotometer. Generally, this UV visible spectrophotometer is useful for the determination of concentration of any solvent or the media, but they determine the concentration of this minute and the microfilaments and the microstructure, the nanoparticle, in a different media or the base. So these are our PG students. Now you can see these are again our PG students from repertory department. So they prepare nanoparticle by chemical method, because while preparing nanoparticle there are different procedures, there are different methodology through which we prepare the nanoparticles. So one of the method is chemical method. By the chemical method we use the various chemical ingredients like potassium ferrocyanide and sodium benzoate. So these are the different chemicals which are used to synthesize the minute and the microstructure, which are nanoparticles. And afterward, they standardize it by first UV visible spectrophotometer, second by SEM, scanning electron microscope, and third by the AFM. If it is powdered, so definitely AFM, that is atomic force microscope, plays a major role. It gives you the surface morphology. it gives you the surface properties of that minute and the microstructure. So this again AFM also available in our Parul University in Parul Micro Nano Lab, and we used to have this standardization and the method of preparation with the mother tincture and ultra high dilutions also. So this is our way of working. We established this work with our PG students, with our UG students, to facilitate other peoples also, other faculties also, and to have a right propagation in the field of evidence-based medicine. Because ultimately our path is not evidence-based medicine. Now it is said to be a pseudoscience. It is said to be a alternative medicine. In Wikipedia, you can see, you can search homeopathy. So it is not evidence-based or mainstream medicine. It is alternative or the pseudoscience. So why it is pseudoscience? Because ultimately in our medicine, we are unable to showcase this mass particle. We are unable to showcasing the absorption of this mass particle. We are unable to showcase the distribution of this mass particle. We are unable to showcase the rate of metabolism of this mass particle, because there are different phases of metabolism: phase 1, phase 2, phase 3, phase 4, and we are unable to showcase the distribution of this mass particle. That's why in pharmacology, when we talk about pharmacology, so they have two branches: pharmacodynamic and pharmacokinetic. So what is pharmacodynamic? What the drug does with the body. And what is pharmacokinetic? What the body does with the drug. That is the response of body against the drug. So ultimately we have no PD and PK model. It is again a question mark. We always talk about vital force. We always talk about the vitality. We always talk about the gyroscopic model. But where is the evidence? So we have to prove that. When we prove this modus operandi, so this will be our first Nobel Prize from the field of homeopathy. Afterwards, homeopathy accepted worldwide. So we have to focus on that. Today it is not only just a few slides or the presentation or a session. It gives you a very strong impact to make our homeopathy as evidence-based medicine. Because ultimately in our medicine it is already ultra high diluted. It is above the Avogadro number. It is above 1 into 10 to the power minus 9 and 1 into 10 to the power minus 7. So it is very difficult to showcase the absorption, the rate of metabolism and the excretion and the distribution of this particle, even though they are not particles. They are said to be a memory of that particle. So what we are going to enhance, we are going to create some mass in our ultra high dilution. Afterwards, we go for the standardization. Afterwards, we go for the preclinical trial, because we also want to check the efficacy of these minute structures. So we have in vitro study, we have in vivo model, we have animal model. Afterwards, we should enter into the clinical study. So this is the what we can say a pattern or a sequence to facilitate such type of research work in our PG and the UG students and PhD students also, because we also have PhD in our Parul University. Now this is another faculty from Department of Practice of Medicine. She started preparation of nanoparticle in folliculinum 200C in centesimal scale. So what she prepared this folliculinum nanoparticle with the sugar of milk, because this folliculinum 200, it is again ultra high diluted. So when we standardize it, when we go for their PDPK model, so it is again question mark. So what we are going to start it, we started the synthesizing of some mass particle in such ultra high dilution. This is another work completed by our faculty, Dr. Jayashree Madam from Department of Practice of Medicine in our Parul University, with the help of electric triturator machine. In image you can see this is the electric triturator machine. So she used sugar of milk as the base. Now this is our rate of absorption. This is the determination of arsenic album 200 nanoparticle in human blood serum. Now you can see in this image, this is just like a star shape or a what we can say, the scattering of light from the cluster particle. So this is the structure of arsenic album nanoparticle in 200, which is detected by human blood serum. Now, this is our inauguration of Home Research Unit. We inaugurate this unit in 2024, and after inauguration of this research unit, it is not just a room or a platform. It is the inauguration of our research aptitude, inauguration of our research thoughts, inauguration of our ideas, inauguration of our feelings, and our research mind towards homeopathy. So now we established it in our PIHR, Parul Institute of Homeopathy and Research, which is a part of Parul University, and we started publication of many articles in the peer review journals and in Scopus in Web of Science also. Now, this is our faculty development program. After this inauguration, we started this faculty development by making the bond and the collaboration with the multidisciplinary, with the discipline of repertory department, discipline of pathology department, discipline of materia medica department, discipline of pharmacy. So we have this interdepartmental exchange program. So this is again very fruitful for us, and we started this work. This is our faculty development program in pedagogy session. Pedagogy session, it based on the advanced teaching learning program. So on that particular session, we are having such faculties, and we are having such broad mind spectrum to discuss about the research work, to discuss about the publication, to discuss about the students research and STSH programs. Now you can see this is the in vitro study of the nanoparticle in the oil base. So this is Candida albicans. So this is THB, that is Thuja hair oil from BHADORIA with the nano preparation. In 50 microliter, they are having no zone of inhibition. So it is no result. Whereas in 100 microliter, it shows zone of inhibition, which is around 3.7 plus minus 0.26. So it is the mean value of 3.6, 4.0 and 3.5. So in 50 micron, there is no zone of inhibition. Whereas in 100 micron, you can see the zone of inhibition, which is around 3.7 plus minus. This is the efficacy of nanoparticle in the oil base. Now, this is the determination of nanoparticle, preparation of nanoparticle in the green synthesis. What is green synthesis? We took the Thuja occidental mother tincture because the color of Thuja is dark green in color. They are rich in the heavy alkaloids and the metabolites and the what we can say, the compositions, because they are from plant group. So this Thuja occidental, it is our base for synthesizing the nanoparticle by human blood serum. Now you can see these are the nanostructures, nanoparticle, which are prepared by toxic human blood serum because we mix alcohol in the blood. So this is a toxic human blood, and the main vehicle is the Thuja occidental mother tincture. So it is said to be a green revolution or the green synthesis. So you can see the different pattern of arrangement, different shape, different size, different structure of this toxic human blood serum nanoparticle with the Thuja occidentalis mother tincture in the hydroalcoholic base. Now this is the standardization. So after preparation, we started standardization first by organoleptic evaluation, by their color, odor, taste, texture, fractures. So these are the organoleptic evaluation. In botanical aspect, we have both macro and micro. So in both macro and micro, we are going to determine their shape, their external feature, their quantification, their qualification by scanning electron microscope or transmission electron microscope also. Whereas in the physical property, we have this moisture content, extraction value, ash value, fluorescent test. So these all are regarding with the physical parameter. In the chemical, we have this chromatography, HPTLC, GC, GCMS, DNA fingerprinting. These all are chemical properties for such minute and the microstructure. In the biological evaluation, we go for in vitro study, in vivo study, microbial contamination, and antagonistic. In previous slide, I already taught about you the in vitro study in couplet method, Candida albicans. So this comes under biological evaluation. So just to prepare the nanoparticle is not our main aim. Our main aim is to show their efficacy in the biological model. So this comes under the major part and the major role of the nanoparticle. Another one is the pharmacological aspect. The pharmacological is the bitterness value, the hemolytic activity, foaming index, swelling index. So this comes under the pharmacological activity. And in toxicological aspect, we have animal model, we have in vivo model, we have animal house. So by different species like rats, mice, rabbits, so we determine the toxicity of such nanoparticles. So before preclinical, we have to determine the toxicity, we have to determine their hemolytic activity, we have to determine their foaming index, and afterward we enters into the clinical model. So these are the major aspects for the standardization of such micro and the minute structures which are said to be a nanoparticle. So this is all about today's presentation. It is not much more sufficient, but we just started this work and this idea with our platform, and to discuss about what are the nanoparticles, what are the different methods of preparation, what are the different properties of nanoparticles, what is the history behind it, and what are the different ways of talking about the nanoparticles. So this is all about today's session. In next session, we will discuss about the various methods in our homeopathy, how we will prepare this minute and the microstructure, the massless particle in ultra high dilution. So in next lecture, in next time, we will definitely discuss about this, and I will definitely share this PPT to Lavanya side for this handling and for showcasing this work, and to share this work with other people also. Thank you so much.
Thank you, sir. Thank you so much for your knowledgeable lecture, and we are so overwhelmed to learn regarding the nanoparticles also. So today we have our one of our guests, sir, Dr. Niveta Patnaik, ma'am, our National Vice President of AIHSAYA, sir. So over to you, ma'am. Dr. Niveta, ma'am. Am I audible to you, ma'am? Dr. Niveta, ma'am.
Yes, audible, audible.
Good evening and namaste to everybody. And Dr. Suraj, thank you so much for your so important. I have completely listened your completely presentation. It was too good. Heartily thankful to you for your presentation and give us so much knowledge. Really, I am so glad that in homeopathy, like you, the homeopaths, matlab nowadays they are so much advanced rather than just like therapeutic homeopathic system of medicine, rather than the scientific way, completely based on the how it is based on the nanoparticles. Really, when I came to know your topic is based on the nanoparticle in homeopathy. I told to Dr. Lavanya, please post me the link, because I have to post in all my classes, and also in PG and PhD students. I don't know they have joined or not, but I'm so happy, heartily thankful to you, Dr. Suraj, and Dr. Lavanya, my little darling. Thank you so much to inform me regarding this. And I was so glad. I was so glad uh, that I have listened and I joined in this program. Thank you all. And Dr. Kingsuk, love you, beta. uh, Everything is going on very smoothly. So happy for this. And also, again, we'll organize the seminar and we'll inform particular to all the speakers and the students and doctors also. That is also my responsibility. Uh, for this, I am heartily thankful to you all. Thank you, dear. Okay, bye.
Thank you so much, ma'am. Your words are very important for us. Thank you so much, ma'am. And we are always ready for this type of work, for sharing and for this facilitation. Thank you so much, ma'am. Thank you, Lavanya. Thankful to all team members of this platform.
थैंक यू सो मच डॉक्टर निवेदिता मैडम। अरे मैडम, आपका आशीर्वाद है हम लोग के साथ, तो हम लोग बहुत बहुत काम कर सकते हैं मैम। सिर्फ आपका गाइडेंस चाहिए, ब्लेसिंग चाहिए हम लोग को।
Thank you.
मैडम चलिए एक हम लोग ग्रुप सेल्फी ले लेते हैं।
अच्छा अच्छा।
फोटो ऑन कर मिलते हैं अगस्त को।
Thank you so much, sir. Dr. Suraj sir, Dr. Kinshuk sir, Dr. Nivedita Patnaik ma'am. And a special thanks to Suraj sir. We will looking forward for the many more sessions and keep on going for the research work, sir. Yes, sir. Yeah.
Thank you to
So,
all.
respected participants, we'll be moving on to the day after tomorrow session. So, tomorrow there will be no session. And on Saturday there will be an exciting session. So, stay tuned. Yeah, we'll be right back soon. Yeah.
Okay, okay. Done. Bye.
Thank you so much.